Hey everyone, so today we're talking about the cell cycle and checkpoints. We're going to get into a little bit of prophase, metaphase, anaphase, telophase, typical mitosis stuff, though that is going to be a separate lecture. Let's get into it. So we're going to be talking a little bit about some enzymes that are really important. So I wanted to cover one of these. We're going to see it a bunch because kinases are integral to cell signaling in general, but especially signaling during the cell cycle. And so I just wanted to talk a little bit about kinases. They phosphorylate. So that means that they take a phosphate typically from ATP, though they could also take it from GTP, rarely CTP or TTP. Those can exist, but they're pretty rare. They're not typical signaling molecules. We do see GTP more, so that's a guanine instead of an adenine. But for the most part, what kinases are going to do is they're going to take a phosphate from ATP and put that onto a substrate. So that's just whatever protein or sugar, it's usually a protein, another enzyme that they're acting upon. So that's the substrate. And so that's what kinases do. They phosphorylate. And one of the key things to keep in mind is that the kinase is going to phosphorylate a substrate. Right, so it's substrate. But this enzyme right here could also be a kinase. And so what we see is when we phosphorylate something, oftentimes that's kind of like taking a battery from the ATP, and we're taking that battery and putting it into the enzyme to start it working. And so it's slightly more complicated than that, of course, but honestly, that's a good way to think about it. Like when I phosphorylate something, I'm turning it on. So I'm getting it to do its actual job. Now, the cell cycle, you may have encountered this before, you may have not. It's pretty straightforward. It's all the events that have to occur in order to get one cell to form new daughter cells. And so it's pretty straightforward. I'm going to be approaching it from a eukaryotic perspective. Bacteria kind of engage in similar strategies, except we would call that like binary fission as opposed to mitosis, but it is relatively similar, but we're talking eukaryotic cells here. So we do have all these different stages. I'm going to go through each of those stages, and each of those stages, pretty much each stage has checkpoints. And those checkpoints are going to restrict excessive growth. Okay, and so I'm going to spend a whole bunch of time talking about those as well. Now, if you're not in your resting G0 state, or if you are or if you're not undergoing mitosis or meiosis, so if you're not in your M phase or your G0 phase, then we call all the rest of this, so the blue, the red, and the green here, those are called interphase. Okay, so that you got your G1, your S, and your G2, your gap, your synthesis, and your second gap. That's what we call interphase. So you could say G1 interphase or something like that. So that's kind of where that nomenclature comes from. Moving on to, we're just going to go through each of the interphases. So the G0, it's also called the resting phase, or it can be called quiescent phase. That's the normal state for most cells that are not actively replicating. And it kind of depends on the cell type of what we call it. So it can be reversible, it could be irreversible. If it's irreversible, we call that like its terminal state. So we see that on a variety of cells. So you think about the human body, what kind of cell types don't typically replicate? So neurons come to mind. A lot of muscle cells are kind of terminal. They themselves don't replicate. It's the stem cells that will become muscle cells. Those are where the actual replication occurs. So it depends on cell type. But if you're just kind of stuck in the G0 phase, that's fine. You can just come out of that. And that's I have a few other slides to talk about how we actually initiate exiting the G0 phase and entering the G1 phase. Now, there are some cells that actually never fully enter the G0 phase. And so, like, you think your epithelials. So if you get a cut in your mouth or like you bite your cheek or something like that, it heals really fast, right? Or all the epithelials that line your entire digestive tract, or even endothelial cells, like in your blood vessels. Those repair really fast. They're ready to go. And so sometimes they can be sitting in, it's arguable whether they're in a quiescent state or not. Or if they're sort of in an early G1, they kind of hang out here. So it's sort of hard to tell, but basically they're ready to divide. They never really exit the gap phases. They never exit and go into the G0 phase. Okay, so let's talk G1. So our first gap, otherwise known as growth, the G technically stands for gap, but growth is another great way to think about it. So this is the largest phase, obviously. I like this diagram a lot. You can tell there's a lot more going on. We're going to see more of this diagram later. So I have it kind of blocked out here. You can't really tell, but there's a lot of things hidden underneath this diagram. And so you can see G1 is the biggest. That's when we're going to get ready to make two cells, right? So if you need to make one cell into two cells, you got to double everything, right? If you were living in a house and you want to move into a second house right next door and keep the same amount of stuff, you're going to need two couches, you're going to need two TVs, you're going to need two beds or whatever. So you have to start buying things. So you have to start building more organelles. So if you're going to have two daughter cells that look exactly the same to the first one, well, you're going to need twice as many mitochondria. You're going to need twice as many ribosomes. So you get the hint there. You're also drastically increasing in size. Not just so something I wanted to emphasize was that the physical size of the cell does increase, but also just the numbers of membrane-bound organelles, your Golgi apparatus, your ER, those are going to increase as well. And so not just the physical size, but the amount of lipid bilayers that are just present inside the cell is going to increase a lot. And to kind of pay for all of this, you're going to have increased metabolism. And along with that comes protein production. And so my issue with a lot of diagrams that you'll find or lectures that talk about this is they gloss over a lot of things. So I'm going to try and dig a little bit deeper. So they'll, a lot of times when you read about gap phases or whatever, they'll say the increased protein production. Oh, okay, well, yeah, duh. It's making more mitochondria. It's making all these other things. Like, yeah, you're going to have increased protein production. Well, what does that really mean? Well, we need to talk a little bit about that. But to give you just a smattering, there's kind of the level of stuff that you need to understand, and then there's the level of stuff that's actually kind of going on underneath. And as you know, in this class, we're going to dig through a few layers. We're going to peel back a few layers of the onion that is biology, and we're going to find, some deeper stuff. But you can always keep peeling back layers, since I'm going to kind of show you where you could keep peeling layers if you wanted to. It is the longest phase, the gap one, and it has the first major checkpoint that checks if there's any DNA damage, and essentially is there enough material to go into the S phase. And we're going to talk more about checkpoints and how we actually enter different phases later on. So I want to just kind of cover and give you a general synopsis of each phase, and then we'll dig a little bit deeper, okay? Now, as we enter the S phase, so our last lecture was on DNA replication. And this is where that occurs. So when, if we kind of zoom out a little bit, DNA replication, you got your DNA polymerases, you got your topoisomerases, you got your helicases, all that crazy stuff. That's happening in a human and thousands, tens of thousands of different locations on the chromosomes. That's happening during the synthesis phase. So that's when we duplicate our chromosomes. That's going to result in sister chromatids. So if I have, let's say this chromosome was from dad, and this chromosome was from mom. Let's say it's, the 13th chromosome. So we've got one from dad, one from mom, and this is like you. And then we need to make more cells. So we're going to undergo replication during the S phase, and then I end up with two sister chromatids. So there's one sister from dad, one sister from dad. There's one sister from mom, one sister from mom, and they're connected at the centromeres. And those are going to come up later. The actual kinetochore, which is where our spindle fibers are going to attach to pull apart during anaphase. They attach during metaphase and pull apart during anaphase. We're going to talk about the centromeres, and that's a really, really key checkpoint. But there is another checkpoint that occurs, so that checkpoint happens during M, but this checkpoint occurs here just to check for DNA damage. So a lot of times you think about when we undergo DNA replication, your actual DNA polymerases do some fact checking, right? They do 3 to 5 prime checking, and they can fix those problems. They fix a lot of problems that way. However, you can have damage occur in other ways. So like X-rays or just other radiation, gamma radiation, trying to think what else, toxins can cause DNA damage. And so here is one of those places where you're going to actually look. And I have a whole slide devoted to how we actually, the pathway involved with checking for DNA damage. It's actually really complicated and one of probably the most poorly understood of all the checkpoints, but we do know it does check for DNA damage. Something really important here, whenever you see these depicted, so when we have the S phase occur, we double our DNA. Okay, awesome. We usually depict the chromosomes this way as condensed. That is not what they look like. They're actually the most loose that they've ever been during the S phase. They are not condensed. So they don't look like this. They kind of just look all spilly outy, like they're just kind of everywhere. Because you want your DNA, if it's undergoing replication, to be kind of loose. You don't want to start running into like these dense areas with your DNA polymerase. So you want it to be nice and loose so that you can actually do the replication. Okay, so we usually depict it this way because that's the easiest way to visualize it, but it doesn't actually look like that. This condensation doesn't actually occur until like prophase. All right. So then we move on to the second gap phase. It's the shortest one. They're showing it, it's decently large here. I mean, there are some like cancers that just straight up skip gap 2. This is just so you can really see it. It's super small. Honestly, S phase is actually relatively small. The G1 is probably like more like 90% of this. You can have a little bit more growth. Some diagrams will depict this as like, oh, it's the second growth phase, so you're going to get all kinds of growth and stuff. Honestly, you should have your organelles all preset. You should have your proteins ready. You're going to have a little bit of increase in all of those things, but for the most part, it's really for the third checkpoint, which you're going to do one last DNA damage check and repair. There are actually 2 checkpoints that occur here, and we're going to talk about those later. But that's the main point, because once you start entering mitosis, I mean, you're kind of stuck going through that whole process. So you better make sure you're ready to go. All right, so then we enter our M phase, which is mitosis or meiosis. We're actually going to spend 2 whole weeks talking about those 2 things. You've probably encountered it before. You got your prophase, metaphase, anaphase, telophase. Some people include like a pro-metaphase. Something to keep in mind, I mean, we're going to talk more about this later, but remember that it's one continuous flow from interphase, which is where we are from our gap phase, all the way through 2 new daughter cells. So we delineate those things, right? So we say, oh, this is prophase, and this is metaphase, and this is anaphase, but it's kind of a continuous process. And so just understanding that is really important. All these biological processes are, for the most part, like that one continuous flowing process. They're not these very hard set things like, oh, okay, this is exactly when metaphase is. Well, it can be kind of tricky, hence pro-metaphase. Now, I also, there's another checkpoint here. I say the 4th checkpoint. In general, it kind of depends. There's more than 4, but I'll get to that later. But this is when you have the mitotic spindle fiber attachment. You ensure that during pro-metaphase and metaphase, you actually get the spindle fibers attached to your sister chromatids at the kinetochore in the centromere. And so that's where that occurs. And we're going to have to talk about that later. So let's actually talk about these checkpoints. So I said, oh, that's the 4th checkpoint. Well, there's actually more like 3, like 6 checkpoints. It depends on who you ask. A lot of diagrams will depict only 3 checkpoints, which I always disagree with. I think there are at least 4. It's arguable that there's 6, but a lot of the checkpoints, there's some similarities, and so that's where some of this gets a little wonky. I mean, you can see there's a DNA damage versus a DNA replication checkpoint, so a lot of the mechanisms to actually control this are similar, and so they use the same proteins to check for the DNA damage, for instance. And so if you go looking for DNA damage and you see, why would you use different proteins to check for the same problem, right? So there's the same checker, but at different time points, basically. This is really, really important. So if you start messing with your checkpoints, then you can get cancer. And so generally, cancers arise for 2 reasons. You have mutations that occur just randomly, or maybe you got exposed to radiation, or so it's something like that causing mutations in these checkpoints. That's usually where it's going to happen. And it's usually in what we call tumor suppressor genes, which I'm going to show you a picture of what I'm talking about. They're the genes that control whether or not you can keep going into the cell cycle. So you can either have that, so just random mutations in those proteins, or you could have passed down generationally mutations. And so that's when you can get mutations like in the BRCA gene. So some of you may, if you have a history of breast cancer in your family or other cancers as well, BRCA1 and BRCA2 are genes that are associated with breast cancer. That's what the BRCA, it's like breast cancer associated gene 1 and 2. And you can actually get a PCR test from your doctor, which is way more expensive than it should be. They're charging like hundreds of dollars for PCR. I mean, one round of PCR costs like $1, so they're making out like bandits there. But that doesn't sound like the insurance system. But you can, if you have the mutation in that gene, well, that the BRCA gene actually codes for tumor suppressors. And so if you have a mutation there that gets passed down, you typically live long enough to produce offspring, but near like 50s, 60s, you might develop breast cancer because you have that mutation. And so that's the other way that you can kind of introduce cancers. And what cancers will do is once a cell becomes cancerous, it's easier for it to cause even more damage to the checkpoints and the proteins involved. And so you'll see like, oh, I just happened to get some random cancer, and then if it sits long enough, it sort of like pushes its way and destroys all these other proteins. Cancer is super interesting. Oncology itself is a fascinating world. But we need to talk about the checkpoints, and there's kind of two ways to look at checkpoints. We need everything to kind of allow us to pass into the checkpoint. So we need to build up the proteins that will say, hey, we're ready to go, let's do it. But then you also have another set of proteins that are going to say, hey, hang on, man. And there's kind of a push and pull between those two things, okay? So we're going to talk about that. And that's where we come up with cyclins and cyclin-dependent kinases. So that's the presence of cyclins and cyclin-dependent kinases are what allow us to proceed into each subsequent interphase step. Whereas tumor suppressor proteins are the opposite. They are going to sort of hold you back and make sure that you're checking your DNA, make sure there's no DNA damage and things like that. So let's dig a little bit deeper. And so I've been showing you, I'm just backing up a little bit. So this is what our diagram used to look like, and now it looks like this, much more complicated. See, things are complicated, but it's actually pretty straightforward when you look at it. There's really two things that we're dealing with here, okay? So you can see in this dark gray line here is showing cyclin and CDK. So that stands for cyclin dependent. Okay, so it's relying on the cyclins to bind to it. Kinase. So we talked about kinases earlier, right? So kinases phosphorylate. And we know that phosphorylation typically means that it's activating something. And so what we're going to see is that cyclins and cyclin-dependent kinases, as the amount of them increases, okay, eventually we pass a threshold. And once we've passed that threshold, enough of those cyclins and cyclin-dependent kinases can phosphorylate some of these like deactivating elements that are on the genes that will allow us to basically proceed into the next phase. And so if you look down here, so we have our cyclin and our cyclin-dependent kinase. Ignore all this stuff. We're going to talk about that in a minute. So we've got, in this case, cyclin D binding to cyclin-dependent kinases 4 and 6. And as we proceed on, we pass a threshold. And so what they want to do is RB is a tumor suppressor gene. It's just preventing E2F, this guy right here, from actually proceeding forward and transcribing this gene. So these are the genes that will control S phase. So they're going to allow us to actually start doing replication. So they're going to increase the amount of DNA polymerases that are there. They're going to increase the amount of helicases that are there and all the machinery involved there. And so we don't have that happening. We don't want that to happen until we're ready. And we're not ready until, we don't know that we're ready until we have a bunch of cyclins and cyclin-dependent kinases. And so the presence of these is actually controlled by some other pathways. And so we're going to see that it's all of these just layers and layers of signaling molecules influencing other pathways, which end up signaling another pathway, and then eventually it passes through and we can actually pass through the checkpoint. And so in this case, our cyclin D binds to CDK4 or 6. It's actually both. And then it phosphorylates, right? So you can see our RB here has one phosphate attached, and it gets hyperphosphorylated. Okay, it's hypophosphorylated here, and it's hyperphosphorylated here. And having enough of those phosphates attached actually physically changes the structure of this RB protein and allows it to fall off. That frees this E2F up to actually do transcription of these genes, which so it's going to make RNA, which will then make proteins, and those proteins are going to be involved in the S phase, and that's what allows us to actually pass through here and enter into the S phase. Okay, so it's the presence of cyclin and cyclin-dependent kinase that allows us to pass through here, and we're going to see that throughout each of these phases. So you can see As S phase is progressing, the presence of cyclin A and CDK1 and 2, those are going. If those are high enough, they allow me to proceed into gap2, and the same with cyclin B and CDK1. So you can see that's what's happening, and it's concentration. So you have certain thresholds that you have to meet with your concentrations and of your cyclins and your CDKs. Okay, so that seems pretty straightforward. That's one form of control because the presence or absence of cyclins is also controlled by other factors. So for instance, if I want to undergo cell division, I'm going to send some growth factors. So think a human growth factor, which you might give to like a child who's not growing right or whatever. Um, they said, hey, you could give your kid's kind of short, you can give him some human growth hormone and he'll get really tall. And we said, well, we're both really short, so it kind of makes sense that he would be short, so we're not going to give him human growth hormone. But if we did, human growth hormone is a mitogen. Mitogens bind to signaling pathways really early on in the cell, on the surface of the cell, and they bind and initiate signaling pathways that result in the production of cyclin and cyclin-dependent kinases. And so if you do that enough, if you get enough signaling, then you can pass through and initiate this next level. And then this initiates these. And then you can go to this next phase, and you're looking for more cyclin that gets made by different enzymes. So you can see it gets complicated, but really it's kind of the same thing just over and over again. You're just, what's making the cyclins and cyclin-dependent kinases, and are they there? Do we have enough of them? Are they going to do their thing? Awesome. Now, this happens, great, but that is contrasted with the negative control that comes from tumor suppressors. So tumor suppressor proteins are all this crazy stuff going on here. Of course, there's more than just what's here, but these are some of the biggies. Okay, especially P53. So what these are, we call them tumor suppressor proteins because when we first identified them, we knew, hey, there's a cancer here, and if I throw a bunch of P53 in there, it stops the cancer. Or better yet, I have a cell, if I disable P53, the cell becomes cancerous. So we call it a tumor suppressor gene, or a tumor suppressor protein made by that gene. And those are what control whether or not they can slow down this process. Okay, so they could bind to the cyclins and stop them from doing their job. They can completely stall the process. Or if it gets bad enough, they could actually initiate apoptosis. And so there are lots of these proteins. The most common one that you will hear is P53. It's kind of considered one of the master regulators of tumor suppression. And so you can see here, why is it a master regulator? Because it actually influences pretty much all the checkpoints. All the checkpoints are going to be controlled. They can come down to, you'll see this a lot in pathways, where the pathways, you'll have a bunch of random pathways, like 5 or 6 pathways, but they all end up at the same key protein, and P53 is one of those key proteins, where if you disable P53, all of a sudden you just disabled all of the checkpoints for the cell cycle. So you see in a lot of cancers, P53 is actually disabled, because those cancers, they want to just make all the cyclins they want and not have any issues and not undergo apoptosis, because that means they would die. And so if you disable P53, well, awesome. I can just skip all these checkpoints and just keep on cruising, as long as I have my cyclins and cyclin-dependent kinases. And so that's the idea, this push-pull that can happen. The push comes from the cyclins and cyclin-dependent kinases and their ability to phosphorylate. And then the pull or the stop actually comes from the presence of tumor suppressor genes and proteins. A little bit about each of these phases. So this is the kind of stuff, a lot of times you won't go into this kind of depth. As far as the MCAT is concerned, I would, understanding that cyclins and cyclin-dependent kinases and tumor suppressors, like understanding their functions and each of the gap phases and S phase, like what's actually happening during interphase, those are all going to be key things. And we need to dig a little bit deeper because these are the kinds of questions that I like to ask on the exam, where I could give you a chart like this and then ask you, for example, what if you have excess CDK2 and cyclin A and E, what phase are you likely entering? And you would say the S phase, right? There's CDK2. CDK2 is responsible for 3 different phases, for all the phases, but the presence of cyclin A and E allows you to go into S phase. If it's just A, you would go into gap. If it's D and E, it'd be gap 1. So it gets interesting and complicated, but movement into each phase is controlled by these concentrations of cyclins and cyclin-dependent kinases. Just for the record, when you see things lowercase like this, that means it's mouse. So if it's capital and then lowercase for the rest of the protein or gene, or the mRNA or the gene, then it's in a mouse. But in humans, they're all caps. But they're the same. It's fine. It doesn't matter. A subtlety. But this is really interesting. So I like these kind of charts. You can find a few of these where you see just the rise and fall of different cyclins controls whether or not we're entering each phase. So that's how cells are actually doing stuff, which is really, really cool. Now, as far as checkpoints go, I want to dig a little bit deeper and just talk a little bit about the tumor suppressors. I mentioned these, P53, and we can call those CDK inhibitors. Okay, so let me delineate. There are CDK inhibitors that are artificial, and there are some that are in the cell. They're just tumor suppressor protein. It's kind of a generic term that we use. So they're tumor suppressor proteins that control these massive signaling pathways. Like I said, you can stall it, you can stop it, you can completely destroy the cell. Because sometimes you just cut your losses if you're a cell and you say, you know what, I'm just going to commit suicide because a controlled suicide, which is apoptosis. Because if you were uncontrolled, you could cause inflammation by accident. So that's the big difference. When you undergo apoptosis, you typically don't cause inflammation, which would be bad. So if you're a cell, hey, everything's broken, my DNA is screwed up, and there's no way to repair it. I tried, didn't work, so now I need to just die. P53 is the most important. A bunch of pathways go into P53, but there are other pathways, obviously. Cells are complicated. If you disable these, I already mentioned this, but I don't know, I have a slide for it. So if you disable any of these things, you can have an increase in cancer, or at least the potential for cancer. Now, there are artificial CDK inhibitors. There's a bunch of them, actually, that we use, and those would be examples of chemotherapies. So there are some that specifically inhibit certain cyclins, because a good way to think about cancer, so we say, oh no, I have cancer. Well, actually, to say something like that is kind of generic. That's like saying I had a car accident. Well, there could be all kinds of car accidents. There can be rear ends. There can be front end collisions. There can be side collisions. You could have rolled your car. So cancer is more like 400 or 600 or 1,000 different diseases themselves that all have different hallmarks. Now, a lot of them share similarities. So a lot of cancers will turn off P53, or they will turn off P21 or whatever. And so some cancers have excess cyclins. So if you have a bunch of cyclin, like E, for example, then you're passing through G1 and S, and you're kind of overcoming the potential. Like if you didn't have enough cyclin E, you wouldn't pass through these. And so if you have excess cyclin E, you can just pass through these however you want, and whenever you want. And so if you disable cyclin E, then you can stop that cancer. And some cancers have excess cyclins. Just depends on the cancer. If you want to dig deeper, you can Google it. Now, get ready. This slide's busy, I know, but it's kind of summarizing what's going on. So what I wanted to just show you is the point of this kind of, I have 2 points for showing you this. Number one, it's summarizing what we're talking about. Okay, so if you look at mitogens, that's what I was talking about. I'm going to show you another pathway for this because we need to talk about how do you actually initiate the cell cycle. But so let's assume you get your signaling, and that can go a couple different ways, but basically you're going to go over here. You have your cyclins and your cyclin-dependent kinases here. And if those stimulate themselves, they can make the RB. So we talked about that, right? There's your RB. It's that thing, this guy right here. You can make that fall off, okay? So you can, this means it's stopping it. So that's an important thing on the exams that I make. Knowing that arrows stimulate something and blocked arrows like this are suppressing something, which that's confusing because it's a tumor suppressor, but it's basically like turning that off is what you can think of. But we know that. So cyclins and cyclin-dependent kinases, they phosphorylate this RB, which sort of turns it on, but in doing so makes it fall off, and then it can activate the E2F, and you go into S phase. So really, this is just showing you this exact same thing right here. It just looks different, okay? But down here, we have this part here is this part, okay? And you can see we have our P53 there, and it can stop the cell cycle. And you can have a CDK inhibitor that blocks this, the cyclin-dependent kinases. All kinds of things. Why am I showing you this? So number one, it's reiterating it again. But number two is because these are the kind of pathways that I like to put on exams, and I will ask you questions on them. I will always give you the pathway. I won't say, hey, remember that pathway I randomly showed you in some PowerPoint like 4 weeks ago? you should have memorized it. Now, I'm not that mean. So what I want you to do is just to be able to take a pathway, and a lot of times I like to pick new pathways that you've never seen. They're usually addressing a topic that we have covered, but you should be able to take something new, some new piece of information, like a diagram like this, or a paragraph, or an article, or something like that, and be able to answer questions on it. Because that's what you're going to have to do as you grow up and go to med school or grad school or start some job, or they're going to make you solve problems using new information based on what you already know. So that's kind of cool. But I wanted to talk briefly about how we actually stimulate the entry into the cell cycle. Get to it. I keep saying mitogens, so those can be anything. So I said growth factors, just various hormones. It kind of depends on the cell type. And so you can imagine inside of your body, you've got all these cells. Picture some like a teenage boy or something. He's about to undergo puberty. He's kind of like in that in-between, it's super awkward phase. You're like not a kid, but you're not a teenager. You're like not an adult. It's really awkward. All kinds of changes are happening. And so you've got all kinds of crazy growth factors going on inside your body. Now, depending on the cell type and the cell growth factor, some cells you want to be stimulated by testosterone or estrogen or whatever. Other cells you don't want to be stimulated that way. You want them stimulated a different way. And so you can change the control that is exerted by these mitogens on the cells by what receptors the cells have. Now, those could be extracellular. So in this case, we've got, so imagine here, usually what's happening is you're going to have your phospholipid bilayer for the cell surface is kind of right here. And these are like transmembrane proteins, and then these guys are sticking out from the membrane itself. Okay, so you can see it would be here, and probably like right here, and I think that's kind of what they're showing here for ERB. And so when you have different shaped receptors, those can accept different growth factors, and it just depends on what the growth factor is. But mitogens are typically relatively small peptides or proteins. I mean, not that big. Kilodalton maybe, that seems too big. They're just not that big. Like, let's say 20 or 30 amino acids. And so what that does is these are often kinases as well. And so, kinases, we've talked about that. So when you get the binding of this receptor to its ligand, which in this case is a mitogen, then it initiates a signaling cascade. Now that can occur extracellularly, which is the most obvious thought process behind it. But you can also have intracellular ones. So if you can, insulin's a great example. And so if insulin gets into the cell, that's where it binds its actual receptor, which these are called, they're often called tyrosine kinases because they have a lot of tyrosine in there, but they're kinases. Okay? They phosphorylate something else. And oftentimes they phosphorylate other kinases to activate them. And so just the binding of this is what initiates the signaling cascade. And what that actually does is it amplifies the signal. So for instance, if I get binding here, and it's amplifying this signal here, it's going to go, let's say it can phosphorylate 10 other kinases that are the next step. Well, each of those can then stimulate another 10 kinases. So now I've gone from one signal to now 100 molecules, and each of those can do another 10. And so you can see that's the cascade effect. It snowballs into a massive signaling cascade. And usually what it's doing is it's ending up making transcription factors. And transcription factors are something we haven't talked about yet. We're going to talk about it when we talk about transcription. But transcription factors are like proteins that will go into the DNA and bind to a sequence of DNA and initiate transcription. It occurs when we need to make cyclins or cyclin-dependent kinases or whatever it is. And that's the presence of those things will allow us to then enter into different phases. And that's just kind of how life works. It's great. Now, the binding of those mitogens is what actually triggers this. Mitogens can bind all kinds of stuff. The mito in mitogen actually comes from like mitosis, because we knew you could take this protein, this is like 70 years ago, you could take this little protein and put it on a cell and it would cause it to undergo mitosis. And so they're like, oh my gosh, that's so cool. It's a little protein, so it's called a mitogen. And it causes mitosis. It's kind of fun. Now, they can be surface, they can be intercellular, and I just wanted to point out, so here, would be the first phase. Okay, so we've talked about this already. So there's cyclin D, all right? So let me let me back up a little bit. So there's cyclin D and CDK4 and 6. Okay, so if I jump back, so there's cyclin D and CDK4, 6. Okay, so it's just a different kind of diagram, and that's important too to be able to recognize the similarities and the things that are the same and the things that are different in different types of diagrams. So I like to give you different kinds of diagrams. And so here we have, that's where your CDK inhibitors would bind, or CDKN2AB. I don't know what that is. That's probably what this paper is actually talking about. You can go to this paper down here. I got most of these subsequent diagrams are from primary articles, so like where people like did some research and then made a cool diagram. So they're really nice. And so what I'm trying to emphasize here is there's that RB and the E2F, so the RB gets phosphorylated and falls off, E2F goes on and makes us go enter the S phase. Awesome. We've talked about this a bunch. But how do we actually get the cyclin D there? Well, that's a great question, and it's triggered by these massive pathways. So if you want to dig deeper, you can Google any of these plus the term pathway, and you will get so many pathways. You can do mTOR pathway. That's one of the key proteins that you'll actually encounter in one of these. The RAS or the MEK-ERK, those are huge signaling pathways that a lot of these proteins do double, triple, quadruple duty for different pathways. And so you end up stimulating one kinase, and then it stimulates another pathway and another pathway. And just think about all the different things that need to happen to have a cell divide. It's more than just the presence or absence of cyclin. You need to be doubling your mitochondria. You need to be increasing the amount of phospholipids you have. And so these pathways stimulate more than just this stuff. But what I wanted to do is I added these little labels so you can see how many kinases are actually involved in this process. So I looked up all of these. So actually, some of these are kinases as well. But of all these precursor ones, so your mitogens would bind to like your receptor tyrosine kinases. So that's these. See, tyrosine kinase. So same thing. So receptor tyrosine kinases. That's a kinase. That's a kinase. That's a kinase. All of these are kinases. This one is not. I was very upset to find out it's not a kinase. And then RAS, which is super common, and you can see there's signaling in between these two. RAS is a GTPase, which got mentioned in the primary article previously. And what GTPases are is they use they're little switches, and so they're similar to kinases in that they're dealing with phosphates. But so if you have a GTPase, it's grabbing onto, it's active if it has a GTP attached with 3 phosphates. If it loses one of those phosphates, it gets turned off. So if it goes from a GTP to a GDP. So that's really interesting. So a lot of times GTPases act as switches more so than kinases do. And what actually occurs is that RAS is the switch that then binds to this kinase to sort of activate it. It's complicated. You can dig deeper. It's molecular biology. It's pretty cool if you ask me. So if you want to dig a little deeper. Now, the last couple things I wanted to mention, I want to talk about a couple of the checkpoints because they're really interesting. And there's some cool things that I could ask you on an exam. So this next slide, so you'll get this slide set. I put the diagram that we've been looking at here. So the whole diagram is here. It's each of these, it's just cropped so you can find them on these other ones, but I just put the whole thing here, and the link is down here too if you wanted the link. It's a really good diagram. It's really big. It's like poster size, which is nice because then it looks really smooth in the PowerPoint. But there it is right there for you. And you can see they're going to talk a lot about all kinds of different things. So what's an oncogene? So oncogenes would be like cyclins and cyclin-dependent kinases. So onco meaning oncogenesis, or oncology, so the study of cancer. So oncogenes are genes and the proteins that they make from those genes that we see in cancers. So like if you're a cancer, you need a lot of cyclins and you need a lot of cyclin-dependent kinases. And so if you see a bunch of those, early on we learned that cancers have those, and so we call them oncogenes. Now we understand more the molecular level of like what's actually happening, but we still call them oncogenes. And same with tumor suppressors. So we knew early on that they were suppressing tumors, but we didn't really know what was physically happening, but now we do. So that's really cool. Now, I will give a caveat there. We don't always know exactly what's happening, which is what's really, really cool and why I like science a lot, because there's so many things that we don't know. In case in point would be the S phase checkpoint. So I tried to do some digging. So we know that there's a checkpoint during the S phase. And we know that it is responsible for looking for any damage. So that can be from radiation, it can be from ultraviolet light. So basically, you're looking for DNA damage, which we will be covering later on when we talk about mutations and mutation repair. Those are two separate topics. This will come up again. And so you can see, I love these kinds of diagrams because they have just giant question marks because this is active areas of research. We don't know what happens. So is it a template switch? Is it reverse and repair? It's probably both, knowing how cells work. And so being able to like delineate and test these things, that's science, right? So there's a grad project. Figure out if this goes this way or this way. That's a giant question. So that could be an entire lab's work of work to actually figure that out. So, but anyway, so the S phase checkpoint is complicated. And so I'm not going to go through this because there's a lot going on. There's not much to talk about. I mean, you can just see the pathways. But this is the kind of thing I could give you a pathway like this and then say, okay, well, tell me if I broke ATR, what would that do? And so if I disabled ATR, what would that mean? Oh, well, then this isn't going to work, and then this whole process isn't going to work, and you're not going to slow the replication. It would go faster. So I could say, if you disabled ATR, what would happen to replication? You would say, well, it would go fast because this isn't happening, so you're not going to slow it. You're going to make it go fast, or whatever. So I like those kinds of things. So that's the S phase. This article is really interesting, talking about S phase. We don't know. There's a lot of things about the S phase checkpoint. It's the least understood of all the checkpoints. The other one I wanted to mention, so just backing up a little bit, there's 4 to 6 checkpoints. It all depends, but most of them, so let me back up here. So most of them involve just looking at the DNA itself. Are there single-strand breaks? Are there double-strand breaks? Are there nucleotide problems? Do you see thymine dimers? All kinds of stuff that we're going to talk about with mutations. Do you see those things? Okay, awesome. Now, the S phase check is a little bit different. Okay, so it's still looking for DNA damage though. But then we also have some other checks, and the main one that I want to mention now was the spindle assembly checkpoint. Okay? And that's what occurs during mitosis. Okay, so all these other ones usually involve tumor suppressors. So they're almost all the same signaling pathway. So like if you look here, we're going from, there's P53 and P21, there's P21 again, there's some other tumor suppressors, there's P53 again, there's P21. And so you can see a lot of these checkpoints share similarities, but the M phase checkpoint is different. Just like the S phase, it does not involve those same ones. And the M phase spindle checkpoint does not either. I know this is kind of a busy slide. This paper was awesome. It's one of the best papers I've seen in a long time. If you are interested in understanding everything we know about M-phase spindle checkpoints, which, I mean, I know you've been clamoring for that, you can head to this paper and read. They have tons of really cool pictures like this one. Like, this is a really good picture. There's like 6 or 7 pictures like this that talk about all the cool molecular subtleties. So I recommend going and checking out this paper. It was really good. And so basically, we know that, so prophase, we have our chromosomes kind of condensed down. Pro-metaphase, they're starting to get attached here. So that's kind of where we're looking. So before we enter metaphase, we want to make sure that our spindle fibers, okay, which are a series of microtubules. If you take cell bio, we're going to talk about that. So there's microtubules, there's actin filaments, there's just all kinds of, the cytoskeleton of the cell needs to attach to this, the kinetochore here. Okay, so the centromere is like the central area, but the kinetochore is the actual like physical like chunk here. So you can see centromeric chromatin. So it's just, the centromere is more like the DNA portion, and then the kinetochore is like the proteins associated with this little area. And so what we want is we need to make sure that our spindle fibers are attached because we're about to pull them apart during anaphase. So if we're going to line up our things during metaphase and pull them apart during anaphase, we need to make sure that even to line them up, you need to make sure that your spindle fiber is attached. And so we can see if everything's going correctly, then awesome. So basically you have this, it's the KMN here with this domain, which this is a little more biochemistry, but it's fine. A lot of you are in biochem. But so you can see this protein here. These proteins are going to bind to the microtubule, and that sort of allows this whole kinetochore area to just be kind of sealed off. The microtubule is sealing it off from the rest of the cell, and it's ready to go. So it's sealed. Now, if it's not sealed, so if I don't have the microtubule here, that allows these other proteins to come in and start phosphorylating things. They're messing around with stuff. We don't want them here. Basically, what they're going to do is they're going to come along and say, hey, I have access to this. Sweet. I'm going to break everything and tell the cell that it cannot undergo metaphase and anaphase, okay? Because this guy can gain access here. The process is called the sac. I know. And so if the sac is turned on, oh my gosh, if it's turned on, then that will disable this. So you can't undergo metaphase and anaphase if this little dude is in here, because that tells this other giant cluster of proteins to be turned on and thus not allow it to happen. So this has signaling further on. But basically, if this guy can access this, it's going to turn that on, which means you can't go metaphase or anaphase. Now, if they don't have access to this because this giant microtubule is basically blocking them from getting access, awesome. Then you can undergo metaphase and then anaphase, and it's awesome. Okay, so I spelled it correctly. Now, that's all I want to talk about, but that's kind of a different approach to controlling the cell cycle. So this, we do mention this in our checkpoints, but it's not necessarily super similar to other checkpoints that occurred during the cell cycle.